Okay, so I figured for this video I would go back a little bit about something we were talking about earlier in the week, from the DNA replication and repair PowerPoint to just make sure that we're all on the same page with all of this information. And so as we started talking about back in class, the concept here when we're trying to replicate our DNA. We have two strands of DNA, one that runs from the 5' end to the 3' end, right? So we have 5' to 3', and then one that is 3' to 5'. And the general concept is, again, when we're doing this from the perspective of um, a replication fork. So when we're thinking about this, right, the replication fork or the replication bubble really looks something like so. And so, as we might imagine, if this is our 5' and this is our 3', and this is our 3', and this is our 5', right? If we're working in different directions, again, the DNA polymerase is going to read 3' to 5' because it needs a free 3' end to be able to replicate DNA or polymerize the DNA. So we'll have two different contiguousness strands being formed, again in the 3' to 5' direction, and then 3' to 5' direction. So these two strands that are able to be read through and polymerize, we call them both the leading strand. So, DNA polymerase, it works in the 5' to 3' direction. And so the other strands are where we're going to get these primers or RNA primers that are going to become what we will call Okazaki fragments. And so just as a point of review, right, topoisomerase kind of leads the charge here. It's going to unwind and um, relax the helical structure of DNA. We have helicase here, which is important for breaking the double bonds and opening the helix. And separating the nucleotides, and then we have these single strand DNA binding proteins that are going to stabilize the DNA to prevent it from uh, linking back onto itself. So again, where we have our leading strand, again, when we're making 5' to 3', the actual sequence here is being polymerized by two enzymes. Well, it's being polymerized by one enzyme, that is DNA polymerase. And again, DNA polymerase 3 is the primary polymerase for this function. And where we have the lagging strand, again, that is where the 5' end is is in one direction, and we're basically moving towards the 3' end rather than the other way around. We have primase, which is going to lay down an RNA primer. Now, each RNA primer is not anything in particular in the sense that it's not really big, but it's just enough nucleotides so that DNA polymerase can actually grab a foothold of this sequence in order to continuously or move towards polymerizing the next segment. So as DNA polymerase moves and again works, it's polymerizing in the 5 to 3 direction. So it's reading 3 to 5, right? It's reading 3 to 5. It is synthesizing 5' to 3' along those 5', 3' carbons. And when it creates each of these RNA primer DNA sequences, we call these Okazaki fragments. So again, when we're thinking about this from the perspective of the replication fork opens at the origin of replication, right? And so if we want to think about it from this perspective, this is our, 3' end, this is our 5' end, right? Just like you see here. And so if this is our three prime end on this strand, the the DNA polymerase needs to be working in this direction. However, it can't work in this direction because it's anti-parallel. It's actually now trying to synthesize 5 prime to 3 prime, but in the opposite direction, right? Because the path of the replication fork in this direction is going in this direction, not in the opposite direction. So the leading strand alternates along the origin of replication because, again, if we wanted to separate these two strands, we would say this is our 5 prime end. So this is still our 5 prime end, meaning that this is our 3 prime end, which would indicate that this is now still our 3 prime end and our 5 prime end. So we can read, right? We can read 3 prime to 5 prime, and we're going to synthesize 5 prime to 3 prime. Whereas when we have polymerase moving in the opposite direction, it can't because it's moving against the strain, against the grain, against the strand. So in order to overcome this, this is why we need our primase laying down RNA primer and the ability to polymerize these small segments in between where we have these primers, well, and all of these known as Okazaki fragments. So again, eukaryotes get away with replicating really larger genomes compared to bacteria because unlike bacteria where we have one origin of replication and it just bifurcates kind of in both directions, we have different multiple origins of replication. And again, it's a matter of the process, right? Because DNA polymerase is going to be moving away from this again and to synthesize in the 5 to 3 direction. It's going to synthesize here from the 5 to 3 direction. And the reason that these other sequences are left behind are the ones that lag is because the origin of replication, right? The origin of replication, going back to something we talked about in class, this sequence is going to be what DNA polymerase recognizes as the start point. This is where we're going to begin. So again, it's only going to be able to read in one direction and polymerize in one direction. So these sequences that are left behind are where we now have to fill in the gaps because again, we're now going to be going in this direction for this strand and then this direction for this strand, which is again the opposite direction in which the polymerase is trying to move. So now that we've created these sequences, right? We have these contiguous sequences that are in the leading strands. We have the Okazaki fragments in the lagging strands. But now we have two problems, right? One problem is that we now have RNA, meaning that we have uracil where we should have thymine. And problem two, we haven't created phosphodiester bonds between these different fragments because they're still separated. So how do we overcome this? Well, we overcome this using two different enzymes, one being an exonuclease and one being a ligase. And again, an exonuclease, there are two different kinds of nucleases canonically. There are exonucleases, meaning that if you have a sequence of DNA, this works at either edge of the DNA, like so, or like so. And then you have something called an endonuclease. And endonucleases find sequences that they recognize inside the DNA strand itself. Ligases, as the term might suggest, ligate the phosphodiester bonds that have not fully formed in the backbone of the DNA strand that new nucleotides have been introduced into. So again, DNA polymerase 3, this is our main replicative DNA polymerase. This is going to, again, polymerize in the 5 to 3 direction. When it moves in the 3 to 5 direction, it has the capacity to add on to its function as an exonuclease. So this is how we do a lot of our proofreading. Remember, I told you that we have little in the way of mutations, right? It's about 3 mutations per cell cycle. This is because the same enzyme that does the polymerizing can also do the proofreading. This is also because we have multiple different kinds of polymerases. There are at least five canonical polymerases. DNA polymerase 1 is a specialized form of DNA polymerase. And again, it's also a polymerase that does polymerizing this reaction, but it's also very importantly for the ability to act as an X nuclease for replacing the RNA primers. There are other nucleases which are important for different kinds of damage that happens to the DNA to overcome those, and DNA that's variantly structured. So one of the more common structures of DNA you might find in biology are these nucleotides that look like a big lollipop. These are called hairpins. And hairpin DNA is really hard to unravel and needs specialized polymerases in order to read. So DNA polymerases, and they are going to polymerize this reaction. DNA polymerase 1 is the primary polymerase that breaks down the RNA, and then DNA ligase uh, enters into the fray and reestablishes the sugar phosphate backbone by creating a new phosphodiester bond. So again, we're going to remove the RNA. Again, if we're removing the RNA, we're doing this phosphodiester synthesis along the 3' carbon axis. So now it's about establishing or reestablishing that 5' phosphate, 3' hydroxyl diester bond by losing water. So we're going to lose this OH. This carbon over here is going to lose, uh, this phosphate over here is going to lose an OH. H plus OH is water. And then we end up with a 5' 3' backbone link of a phosphodiester bond. So just some review questions. So we're going to finish this off with DNA damage and repair pathways. So nucleic acids, they are very susceptible to damage, mostly because they are molecules, and molecules are susceptible to damage, not only by way of physical damage, like you see here, but also by way of chemical damage. So chemical modification of the DNA nucleosides can also cause changes to the DNA, such things such as like deamination or the loss of nitrogen species that can lead to point mutations. There's also chemical and exogenous damage to DNA that's physical and chemical, and these damages can occur to the nucleosides here, or they can occur to the sugar phosphate backbone, which could affect single versus double-strand DNA damage. The most common kinds of damage that you see are here. They are single-strand breaks, where either the backbone or a nucleotide is compromised, but it's only one of the two strands that is compromised, and this um, can cause a break or a loss of a nucleotide. This is typically dealt with by a base excision repair. So basically, the damaged nucleotide is removed, so base excision, and a new one is subsequently put in its place in order to complement the strand and seal off the broken backbone. Less common, unless we see this also in a lot in meiosis, is double strand breaks. So this is when two nucleotides typically correspondent with one another or complementary to one another are damaged or removed. This causes an actual physical break in the DNA strand. There's a couple different ways that this can occur for repair. The most common is homologous recombination repair. So this is what we saw before with meiosis crossing over. You have strand invasion here, and then the holiday junction resolves, and the sequence is relatively maintained. Another kind of version of this is something called non-homologous end joining. Non-homologous end joining typically resides in the ability to have a sequence that might end here and another sequence that might end here. And so there's this gap of information. And so we can fill in the gap until we come across a complementarity pair where the complementation occurs. We line that up, and we remove the things that don't overlap or don't fit in the sequence anymore. Now, the interesting thing is non-homologous end joining is actually something that's really important for your immune system. So, how your adaptive immune system functions has a lot to do with the ability that cells have to do non-homologous end joining. The last two different kinds of mutations, which are kind of common, especially when there are high energy or high yield mutation platforms that can lead to higher rates of mutation than breaks of the strand. The first are bulky adducts. And so bulky adducts are molecules that intercalate in the inner section of either the major or minor grooves or actually in the open nucleoside components of DNA. And they are bulky, so they sit inside the DNA, which causes the DNA to bulge. And when we have a bulge that is not at a replication fork, this typically has to be dealt with through nucleotide rescission repair. And sometimes in order to make the DNA come back to its normal orientation, where we have thymines, if we have two thymines that are next to each other, this can be resolved by instead of having the hydrogen bonds between T's and A's, the thymines will actually mobilize these bonds and create something known as a thymine dimer. And when thymine dimers occur, we use something called translesion synthesis, which is the ability to ameliorate this double bond, this thymine dimer, to try to reestablish the connection to the adenines. If not, though, the basic excision repair can actually delete these nucleotides, resulting in a mutation. And then finally, we have nucleotide mutations, substitutions, deletions, insertions. And this is typically what happens as a result of something called mismatch repair. So when we have a new mutation and we get a different nucleotide that was there before, we probably had a C here, and now we have an adenine. We then either allow for this to exist as long as there's a purine pyrimidine match, or the cell will take out this and move in a new nucleotide to reconnect the C to the G, and now the A is over here, by which the cell will actually put in a new T. So we can either get mobilization of the nucleotide, or in some instances, you'll have this as that will become the new the new strand type. So as this gets replicated, you can develop what we call genetic chimerism at this space, where one series of daughter cells will carry the A at this site, and the other will carry the G. So something we've already talked about when we talked about cyclins and types of cell death is the function of P53. P53, again, it is an important protein. It kind of resides in between the function of mitochondria to be able to control apoptotic pathways. It is a stress activated transcription factor that promotes apoptosis when there is a something wrong with the cell. So when the cell recognizes that there's something wrong, P53 gets activated. So if you have a cell, and here's the mitochondria, if you ever have a stimulus, P53 then triggers the mitochondria to release apoptotic markers like cytochrome C. And so P53 in normal times, when it's not being stressed, interacts with another mitochondrial protein called MDM2. And so during regular interactions, MDM2 blocks the ability for P53 to induce apoptosis. But when there is a stimulus, P53 can actually reverse course and cause apoptosis to occur. So apoptosis, again, can be caused by extrinsic or intrinsic signals. The cell will begin by becoming smaller, and it will condense all its chromatin inside the nucleus. The nucleus will begin to bleb, the nucleic acids in the chromosomes will begin to be destroyed and distorted. The cell will begin to bleb as well and begin to come apart. This then allows for the organelles and the membranes to become partitioned inside of various compartments of the cell, and the different compartments of the cell will become apoptotic bodies, which are then consumed by immune cells inside the body, mostly in the form of macrophages and dendritic cells. And now, just as a reminder, right, when we do this, because everything's all nicely bound up in these apoptotic bodies, if you're talking about humans, this does not result in any inflammation and no subsequent response. So when you're talking about cell death, when you're talking about cell death in any type of organism, when it's apoptotic, there's no secondary reaction. There is just the cell dies and is then consumed. If, on the other hand, the cell dies by necrosis or any other type of mediated cell death, this will trigger inflammation in humans, or if you're talking about bacteria, protists, other organisms, can trigger a secondary reaction because then that death cascade is not programmed, but brought on by something else, and the cells will react to that. So when we talk about cancer, right? Cancer, remember, there are two different things that have to happen with cancer. The first is that we have to have um, the block of our um, anti tumor genes. So we have to block our tumor suppressor genes, and then secondarily we have to increase, right? We have to increase the metabolic cascade. So we need to block apoptosis. We need to increase expression of pro-growth or proto-oncogenic genes. And when both of these happen, you end up getting oncogenesis. All right, that's it for uh, DNA repair and replication.